everybody, and welcome to this episode of the Healthspan Action Coalition podcast. My name is Dr. Sanjay Bhojraj, here today with my colleague, Dr. Heather Maine. Heather, great to see you.
Hi.
And we're going to talk about stem cells today. And I know in the literature and in popular press and all over social media, people are talking about stem cells as though they are something that's been around for forever. But we're actually going to explore where stem cells came from, how they were initially discovered, and how a nuclear bomb helped us sort out stem cells. So Heather, why don't you kind of launch us into today's topic?
So I've been working with stem cells for many years. I started back in 2003 and I work with pluripotent stem cells. So the term stem cells is actually one of my pet peeves. When people say stem cells, it's not just one thing. There's many different types of stem cells. And so what we're going to get into today is a little bit about different types of stem cells within the body, like blood stem cells, skin stem cells. And we're going to kind of look at it from a perspective of nuclear bombs. And it's quite interesting that stem cells, while they have always been around per se, they were discovered in the 40s after the nuclear bombs in Hiroshima. And so what people found with the survivors, that people who seemed completely untouched by the bombs, they survived. They had no sort of physical signs of injury, and then they were dying like weeks after the bombs. And it was found over time, obviously it takes time to realize what went wrong, that these people were dying of anemia because their blood stem cell systems had been wiped out. And so in the beginning, it's okay because you've got your red blood cells, you've got your white blood cells, they're all kicking along,
they're already
doing
there
the job
and circulating.
they're supposed to do. They're doing, they're functioning. But when you need to produce new ones, and so in these highly proliferative tissues, like the skin, the blood, the digestive system, you're producing new cells constantly. And so these people that experienced, proximity to the bombs where they survived were not hurt, but ended up dying because it just irradiated their stem cells and killed and wiped out their stem cells. So it's a pretty tragic kind of founding in science that's really how we discovered the identity of the cells that replenish, the tissues in our body. And one thing that I came across when I was PhDing up in Stockholm, my friend Kirsty, who's also from Australia and ended up in Stockholm, was studying nuclear bombs for the sake of looking at cellular turnover. And so I thought today it would be a really interesting discussion to look at, if we're looking towards longevity of wanting our bodies to continue to renew and wanting them to fix themselves and regenerate themselves, what is it that the body does already? And maybe there will be ways that we can try and replicate that. So today we've brought along Kirsty to give a bit of a description of her research and basically just to have a chat about the body and what we know about turnover of cells in different tissues of the body. So welcome, Kirsty.
Hi, Kirsty.
Hello, thank you very much for having
How
me.
many things did we get wrong in that initial description of what's going on?
Nothing yet.
Perfect. Well, I would expect nothing
It's coming.
else of Heather. Yeah. So if anything, I'll be the one that kind of probably says the dumb things. But so a nuclear bomb led. Well, you were studying cell turnover, and this led to the study of the aftermath of Hiroshima and Nagasaki, and that somehow led you then to realize that there are these cells that can help us with turnover, help us heal, cells that had been with us since the beginning of time, but now we're able to study and eventually hopefully use to regenerate and to keep ourselves quote unquote young, so to speak. So is that maybe run us through the story of how that came to pass?
So, I think Heather very nicely illustrated the identification in the first place that we have these stem cells and different niches, different environments in the body where they live and they contribute to the mature cells that we have. And that was all kinds of interesting for many people, because how can we exploit this? How can we use this to aid in repair, to promote longevity, to restore lost function, etc. And so I think there's kind of two schools of thought about that. And one is more, how can we take these stem cells, transplant them in and turn them into whatever we want? And the other, which was what I was interested in at the time, was how can we stimulate this endogenously? How can we get our body to augment this and do even more of this? And in order to do that, you really need to know what the baseline, what is the body capable of? What is the baseline level of regeneration in different tissues in the body? And because most of the methods that exist that we know, most of the sort of the data that we know on cell turnover comes from rodent studies, and given the huge difference in lifespan between the species, is it plausible to think that what we found in mouse translate to an animal that lives, 80, 90 years longer? So we didn't really have much information in humans. I mean, obviously had this, from the work that Heather talked about, but this is not something that you can recapitulate.
No,
you can't.
Please don't.
Yeah, and I mean, most of the methods which we use to study cell turnover in animals are just not ethically compatible for working with humans, giving radioactive traces or
Just
so
blasting people full of X-ray to get to wipe out their stem cells.
Yeah. No
So
go.
why don't we, I know that we have a pretty sophisticated audience, but maybe let's just start by defining what is a stem cell, right? Because we hear about this all the time, and we see Instagram ads and go to this, have them injected in your shoulder for frozen shoulder. But what does it mean? Like what is a stem cell? Just a very basic level.
I'll take that one in terms of it's actually quite boring, really. All a stem cell is something that can make new copies of itself and turn into functional cell types. So stem cells themselves have no function in the body except to make new functional cell types. So if we go for something like a blood stem cell, it makes more blood stem cells, and then those blood stem cells are capable of making functional blood cells. And so typically we say there's 4 different populations of stem cells. The embryonic stem cells, like I've worked with, the pluripotent stem cells, the ones that can make every cell type of the body. If you know much about fertility treatments, when they do IVF, egg and a sperm in a dish, give it a stir. After 5 days, after...
Give it a stir.
5 days after fertilization.
Like you're making cocoa.
Exactly. It's that simple. We're not more complex than that. 5 days later, the embryo is basically the size of a full stop. It has 2 types of cells, pluripotent stem cells and trophectoderm. Trophectoderm goes on to make all the goo that sticks the baby to the body, the umbilical cord, the amniotic tissues. The pluripotent stem cells make the body. So all of us at some stage came from around 40 pluripotent stem cells. And now there's a lot of cell therapy going on around the world. We can put that reference down in the under this one as well. There's 115 clinical trials worldwide using pluripotent stem cells to make different cells of the body, to make neurons or blood cells or skin cells, whatever.
Wow.
So pluripotent stem cells, fetal. So when a baby is developing in the body and starts to form tissues, it starts to form a brain, it starts to form, a blood system, bone, muscles, it goes through transitions of stem cells. So muscle stem cells, blood stem cells, brain stem cells. As adults, we still have those types of stem cells, but at a sort of slightly less regenerative capacity. Like when you're a fetus, you're growing pretty fast. We're not growing so fast anymore. So we still have blood stem cells, brain stem cells, gut stem cells, and they can only make the functional cell types of that tissue, unlike pluripotent stem cells, which can make all the tissue types. And
so they've been we call that differentiation, correct? When they go down a certain line, so that now they can't make everything, but they can make everything within one system.
Yeah, and this is the word potent, potential. So pluripotent, they are they have all the potential, pluripotent.
Got
Whereas
you.
you have adult or fetal and multipotent. They have some of the potential, not all of the potential. And then the 4th population, which is so confused, are the mesenchymal stromal cells. And they're often called mesenchymal stem cells. And they're the ones that are coming from the adipose tissue. They're coming from the bone marrow. And the job of these cells is basically just to keep a nice, happy little environment. And so when you're doing a cell therapy from that type of cell, from the mesenchymal stromal cells, they're sort of having these paracrine effects. They're not going into the body, integrating and making a neuron. They're not going in, integrating and becoming blood cell types or becoming muscle or anything like that. They're going in, they're kind of going up to the sad, immune system or the sad tissue and saying, "You'll be okay, you'll be fine. Do you feel better?"
They're like
And
therapists, therapy cells.
3 weeks later, they're gone. 3 weeks later, they're dead. So they're basically those three populations.
All
so
right.
now
and,
if I come
and
back
Kirsty,
to...
so what does this have to do with nuclear bombs?
So that I don't know, but our work. So when I started, there was a lot of exciting studies coming out. When I say I started my postdoc in Sweden, there was a lot of exciting science coming out, challenging the dogma that I grew up with. When I went through high school and uni, we were taught that the neurons you have in your brain as an adult, the ones you're born with, are the ones you die with, which is why damage to the central nervous system is irreparable and it's lifelong. And that was completely accepted. This is how it is. But then there were all these exciting studies coming through in mice, mostly, where they were actually saying, no, there might be parts of the brain where we can actually make neurons. And then they went further to show, and this is a particular part of the brain called the hippocampus. So it's a part of the brain involved in memory and learning and emotions. And in this part of the brain in mice, they were showing you can actually make quite meaningful numbers of new neurons. And then another study went further to show that if they induced anxiety in animals and you got the animals sort of all stressed as like a model of depression, that the neurogenesis, which is the word for making the new neurons, so the neurogenesis in this part of the brain, it went down. And then if they gave antidepressants, it was restored and the animals were not as anxious. If they blocked neurogenesis and gave the antidepressant, then the animals remained anxious. So there was a lot of excitement in the field that, wow, maybe neurogenesis is happening, it's functional, it's important, and maybe if we can target it in the hippocampus, we've got a new treatment for depression, and we can devise better antidepressants that have less side effects. So there was all this excitement around the field, but we still did not know the fundamental question, does this happen in humans?
So I think I just want to take a pause there. So what you're saying is that the emotional state of the animal actually led to changes, cellular changes in the brain. And by giving then a medication, you could overcome those cellular changes. And that has to happen somehow, because up to this point, we thought what you have is what you have, and you probably kill a bunch of brain cells at university. People know how you do that, right? But you can actually overcome that with medications. That's a big deal. I don't think people realize that we can see these cellular changes that quickly. I mean, I'm assuming this was over the course of days to weeks in these mice. So in humans that have kind of a longer generational time or whatever, it can be then days to months or weeks to months where you can see these structural cellular changes in the brain from depression, anxiety, whatever it may be. But the fact that you can overcome them by growing new cells is a big deal. I think that's really cool.
Yeah,
Yeah,
well,
I've never heard that actually.
it was very mouse-led, but we didn't know how it is in humans. But there was a lot of excitement about it. And also, when you take antidepressants, they don't work like immediately.
Right.
So
It takes
it fit
4
the
to 6
time.
weeks.
Yeah,
Yeah.
it's around the time that it takes for neurons to be. So there was sort of circumstantial evidence that this could be working in humans and this could be good, but we don't know. And so that's kind of where I entered into my postdoc, and I was really trying to consider how can we study this in humans. Obviously, we can't do it in all the ways we do it in the mouse or the rat or whatever, because we generally give compounds that are toxic.
Hmm.
And there would be no ethics to ever do this in humans. There was one study actually where a series of patients in Sweden, as part of their cancer treatment, were given BRDU, which is basically a radioactive, one of the components of DNA. And it's radioactive and it gets incorporated when you are making new DNA. And so they were given that for, I think it was a throat cancer. And when these patients ended up dying, a Swedish MD, PhD actually analyzed the tissue, and you can have a look and see whether or not you can look at the radioactive incorporation into DNA. So in this case, it was given to the cancer patients as sort of part of their treatment. But post hoc, post mortem, he actually took parts of the brain, and that was the very first evidence where you could see incorporation into the DNA of neurons in this hippocampus part of the brain. So that was a study done in collaboration with Fred Gage at Salk, and that was published. But apart from that, and these are like, it was not great post-mortem material. So this didn't, it wasn't a whole cohort. We couldn't say a lot about turnover rates, but it was like the first evidence.
What is almost like a feasibility study?
It's like, yeah, there's something here. So,
right.
so I together with Jonas Frisian, who is a bona fide stem cell professor at Karolinska, we looked at using a very, very out there idea at the time that this is now talking about the nuclear bomb test. So during the Cold War, so the mid-50s to the early 60s, there was a lot of above ground nuclear bomb testing. And during this period, there was a lot of detonations, which in the normal atmosphere, you have cosmic ray proton interactions in the atmosphere as they are. And when they collide in the atmosphere, you get neutrons. And neutrons then collide with nitrogen-14 to give off a proton. And the result is carbon-14. So that's the end of the chemistry lesson. That's going on in the atmosphere all the time.
So it's normally carbon-12, but in this case, it becomes carbon-14.
Which is kind of the concept behind carbon dating, so to speak, right? Like they look at the relative amounts of these concentrations. Not to get super nerdy, but we've got a bunch of nerds that listen to the podcast, so they'll get it.
There is a difference there. So in traditional carbon dating, you're looking at decay. So then when people talk about carbon dating, they're mostly referring to archaeological, where you resolve like thousands of years. And that's because you're working on decay. And carbon-14 has a half-life of just under 6,000 years. So if you're looking at decay, you would never resolve the age of a body in a human lifespan. So it was really the advent of accelerated mass spectrometry, which is a particular kind of machine, massive machine, that essentially takes your sample. In our case, we take the DNA out of the cells and we reduce it to graphite. So it's a pure carbon sample. And then it's comprised of all carbon. Carbon-12, we rate the stable, and carbon-13, and then radioactive carbon-14. Radioactive carbon-14 in the normal situation is like 1 to the negative 12% of all carbon. Very, very low. And this machine is so sensitive that what it will do is it'll take your carbon, it'll split it based on its mass. And so because they're a mass of, 11, 12, and 13, they will be accelerated through these giant magnets at slightly different rates. And then they have detectors at different angles. So they can actually, like you said, count the exact number. So you can get a ratio of the amount of stable carbon, carbon-12, to your radioactive carbon. And so these two things, the Cold War and the above ground nuclear bomb testing and the development of accelerated mass spec, came together to enable us to do this. And so to get back to the Cold War, there was lots of above ground nuclear testing, and that cascade that I told you that goes on normally, where you get, these protons and they're hitting nitrogen-14 and you get C14.
And the neutrons and the C4, yeah.
Exactly. That always goes on. But because of all the above ground nuclear bomb testing, there was a lot of detonations. And the detonations caused massive neutron emissions. So those neutrons went into the atmosphere and accelerated that whole cascade I told you. So we end up with what people in the field affectionately call a bomb spike. And if you look at it graphically, you can kind of have a plot that goes for 4,000 years, where the levels have been pretty stable. They go down a little bit in the Industrial Revolution because we were burning fossil fuels, which is C14 depleted. But essentially, it's kind of 4,000 years of straight. And then you get this shoot up mid-50s when the above ground nuclear bomb testing started. And that's the spike. And then in 1963, there was a test ban treaty signed, a worldwide test ban treaty, which was aimed to limit above ground nuclear bomb testing. And so then C14 values were decreasing exponentially ever since. And it's not because of decay. It's not its half-life is, like I said, almost 6,000 years, but it's going largely into the water system and the upper biosphere.
Hmm.
So what you end up having is like a date mark. So in the atmosphere, there's at any given time, there's a rather exquisite ratio of radioactive carbon to stable carbon. And so if you're able to take a population of cells, in our case, we took neurons out of the brain from deceased people. And if you take those neurons and then you isolate out the DNA from those neurons, and then you reduce that DNA to graphite and you measure it using this accelerator mass spectrometry. So it tells you exactly how much radioactive to stable carbon you have in the DNA of that neuron. You can then say how old that population of neurons are. That was the idea. That was the big kind of crazy idea that we.
Wow. So for those of us who may not have the molecular biology background, so DNA is made up of base pairs, and we know this, but those base pairs then are made up of carbon and nitrogen and oxygen and all these things. So you are then looking at the molecular amounts of 14 versus 12 carbon, able to quantify that. And based on this timestamp, when you saw this huge increase in C14 in the environment because of the radiation testing, you were able to then work back the timestamp of, are these new cells or are they old cells, based on how much C14 was incorporated into the DNA. Is that
Exactly.
is that the Cliff Notes version?
That's exactly, because if basically when a cell is born, when a cell is made, its DNA is synthesized, it gets built, a whole lot of building blocks, of which many of those building blocks are carbon. So once it gets made, if it was made before the bomb testing, so say you're born in 1940, you make all your cells, there's no bomb testing at this point. So all your DNA will be devoid of these high levels of carbon 14. And if that cell lives for your whole entire life, it will never incorporate any of the radioactive carbon into its DNA.
Right.
But if you make new neurons sometime after the bomb testing, it will have this radioactive carbon in its DNA. And so if you take, say in our case, we took different parts of the brain, isolated out the neurons and took the DNA, and we took people born at all different ages. And if we took people born way before the bomb testing and they had radioactive levels of carbon in the DNA of their neurons, we knew they had to be making those neurons as adults. So it was proof that adult humans can make new neurons.
So I mean, wow, to get from point A to point C, that sounds like a lot of things. So as you looked at these brains of just civilians, right? These are not people these were just people that existed on Earth during that time. Did you see, and you may not have been able to study people from different geographies, like how close you were, or was this just stuff in the atmosphere, number one? And number two, then clinically, as you're looking at these ratios of 14 to 12 carbon, were there different areas in the brain that had differential amounts of C14, meaning that some areas of the brain clinically are more plastic or more likely to be able to regenerate brain cells than other areas?
Yep. So the first question, geographically, so the C14 globally equalized very quickly. So even though the majority of the above ground nuclear bomb testing was in the Northern Hemisphere, it pretty much equalized around the whole Northern Hemisphere. We use reference curves that tell us the levels of atmospheric carbon 14. And we use the right hemispheric latitude for the curve that we use, because the Southern Hemisphere, so for example, we'd be rather confusing, Heather, because we're born in Australia, our neurons were like wired in Australia, but we're in the Northern Hemisphere. So in the Southern Hemisphere, the peak of the bomb testing is a little bit lower. So there is a slight difference, and we use that when we do our modeling, but it's
Okay.
very much a global effect. It's not just around the within a location of
Right. It's not like within a blast radius of 10 miles or something. So
no,
it
no water.
equilibrates over the hemisphere or different bands of latitude.
Yep. And
Okay.
the second one, yes. So the first thing we looked at was, I mean, this what I explained to you now took me about 4 years to get it to work.
One sentence encompasses 4 years of your hard work. I love it. Love it.
And in the beginning, we looked at the cortex, and we looked at the cerebellum, different parts of the brain, and we saw no evidence for turnover. So that fit the dogma. Basically, what you're born with was keeping with you for life.
Yeah.
And then we looked at the two parts of the brain that nearly every animal looked at has been shown to have this neurogenesis is the hippocampus, which I mentioned, and the olfactory bulb. And olfactory bulb is one of the major sites of neurogenesis in most animals studied. But sense of smell is supremely important to a mouse to identify its young. And the moment it smells something wrong with its young, it kills it. So, and when they're pregnant, they start making a lot of neurons in this part of the brain. So it's really clever. Like they up the neurogenesis so they can heighten their sense of smell so they can identify their young. And that's crazy important if you're a mouse. Not so important if you're a human. So we're not so sure, do we have it or not? So I looked in the olfactory bulb and we didn't find any evidence of neurogenesis, which went against everything, including primates. So we were very, very surprised by that. And I remember visiting a neuroscience department in Israel, and so many people, particularly I remember one PhD student was so depressed. He said, so basically my PhD is for nothing.
That's got to feel demoralizing, I'm sure.
So that was yeah. But then the hippocampus, we do. And actually we could show that even sort of quite far into age. So the hippocampus is a rather big structure. We've got one on each side of the brain. And there's one part in there called the dentate gyrus, and that's the part that's been shown in animals to make new neurons. And it helps us sort of with many tasks, and one of them being, with memory. And they act as like little interneurons talking between other neurons. And so it's thought that their turnover enables higher level plasticity. And we actually found evidence for neurogenesis, and not trivial. I mean, about 700 new neurons added to each hippocampus, so 1,400 new neurons per day.
Wow.
Yeah. It's very controversial. This is a very topic within the field of stem cells and neuroscience. There are very strong proponents that believe neurogenesis does not happen and it's not meaningful. And then there were bucket loads of strong evidence to suggest it does. So this is a field that's got its opinion.
There's fist fights at the meetings.
Yeah.
About
but then,
this.
so this covers the brain quite nicely, but I know that there was at least studies in heart as well, because heart was another one of those tissues where we thought that we didn't have a lot of regeneration.
Yes. Yeah, so this was work that was led by Olaf Bergman and Jonas Thresien, and they were very interested in cardiomyocyte renewal. And so they did exactly what I explained, like what was sort of developed for brain, but then they applied it to looking for markers specific for identifying cardiomyocytes and isolating the nuclei out of the cardiomyocytes, and then doing the same and the DNA dating of that. And they did actually show that it's very low turnover, but there is turnover in cardiomyocytes. And I think across the lifespan, it's about 40% of cardiomyocytes are renewed across the lifespan. So whilst it's a very low amount per year, it's actually not nothing.
Substantial over time.
So it's possible. And, as a cardiologist, of course, I've lived through several different types of stem cell trials where we did crazy things like take a needle into the myocardium itself, like a little tiny cute needle into the myocardium, and try to inject into the adventitia of the cells. And it's difficult to know, and maybe we can back up a little bit, and if you're comfortable, talk about stem cell therapies, because a lot of the stem cell therapy trials have been a little bit surprising, right? We think there's so much promise, and clinically it just doesn't work out. So is it, I guess, a failure of, and I never know if it is a failure of the cells. Is it failure of delivery? Is it just we don't know enough yet? We're just trying to throw, something against the wall and see what sticks. I mean, where have some of these stem cell therapies been surprisingly effective, and where have they been surprisingly ineffective, I guess, as you've grown? Is that
what stem cells are we talking about?
Yeah, well, that's the thing, right? I mean, because that's exactly the point. Is it that we aren't putting the right stem cells in the right location? Do we just not know enough of them?
This is why I say it's my pet peeve of that word, because I've seen some weird studies where, like, you're just putting a certain type of cell into a tissue that just doesn't make any sense. It's not from there. It's not supposed to be there. Like. And then that people are surprised when it doesn't kind of work out. And I'm like, yeah, but what in the world would a neural stem cell be doing in a person's retina? I saw a
Hmm.
study where they put neural stem cells in a person's retina. Like embryonic development puts certain cells in certain places in the body for a reason. And you can't just kind of willy-nilly go taking cells from fat and put them into the heart and think it's going to have some sort of integrating effect. And so I think we have to be very clear on what cell is it and why are we putting it there? Are we putting it there because we want it to turn into a cell type of that tissue? Or are we putting it in there because we want to sort of dampen some kind of immune and inflammatory responses to maybe give those endogenous stem cells the ability to repair the tissue?
Give them some breathing space to do the work that they need to do.
Yeah, exactly. Like maybe
Yeah.
those endogenous stem cells are sitting there, but the environment is so hostile that they're just like, oh, okay, we got
Right.
nothing. So if you make the environment kinder, maybe then they can actually do some natural regeneration. So it's those two things again. It's the endogenous stem cells and the body's natural ability to turn over versus putting in cells that you want to integrate and become functional cells in that tissue type.
Yeah, Kirsty, do you think similarly? Is that as you look at this literature, are we just kind of doing random Frankenstein bolt-ons to tissues that cells don't match?
I'm definitely not versed to talk about stem cell therapies. This is much more Heather's thing. I've always been much more a proponent of promoting endogenous stem cell activity. I mean,
Well, and that leads me to a different question. Do stem cells age? We're talking about embryonic and fetal stem cells, and now I am approaching a half century of age. And are my stem cells the same as 50 years ago when I was born?
at least this is now getting down to definition, and Heather's probably going to jump all over me. So, progenitor, at least we know that progenitor activity decreases. So, at least in the fat, because in making human fat cells called adipocytes, when you make them, these don't divide. These are cells that derive from progenitors, which ultimately come from a stem cell. So, if you sort of have the mothership, the stem cell gives rise to progenitors, which are kind of now they're on their way on a path. They're not going back, but they're still quite plastic. And then they start to fill with lipid and differentiate into a mature fat cell. And so that sort of progenitor pool, it gets depleted, it gets exhausted, and it ages. So therapies that are, there's one school of thought that aging is just about tired stem cells or tired progenitor cells. And if you can kind of get them rejuvenated, you can have a young organ, a young tissue, you can, affect youth in that way.
Yeah. And I think we're going to have a lot of these types of discussions coming up with different companies and different technologies. That's kind of why we wanted to take this moment to define a little bit, what are stem cells and what do we expect from like normal turnover of the body. So I think it illustrates quite well how you might try to drive endogenous regeneration, or you might try to put cells in to drive regeneration. So just so we don't miss anything, we've seen regeneration in a part at least of the brain. We've seen regeneration in cardiac. Did they do the carbon-14 on any other tissues of the body?
Yeah, so Olaf also did it on hepatocytes, so in the liver.
Yeah.
And it's hepatocytes are an extremely interesting cell because the traditional cell, it has, a cell with one sort of nucleus, one center, whereas hepatocytes have the capacity to sort of grow and respond to injury by making two nuclei or keeping one nuclei but putting in twice as much DNA. So they have like amazing ways of dealing with regeneration and injury and growth. And we all know that the liver's got an amazing capacity to renew itself.
Yeah.
So they looked there, and the hepatocytes there very much turn over like every 3 years or something. So it's been used carbon dating for that, and I think confirms what we know as it being a very regenerative and plastic tissue. I did it in the very beginning, crudely looking at intercostal muscle and blood, and what else did I look at?
I would assume bone marrow would turn over.
I tried to do bone marrow, and I was particularly interested in the adipocytes in the bone marrow because they have a big role in shaping a lot of the hematopoietic and like the blood niche, if you will. So But it was really, really important. You need like a lot of cells to make this work, and trying to get bone marrow samples from patients and working with that, I never got it. So I don't have any, I mean, maybe on the whole bone marrow, I was really trying to look at the adipocytes. And I was really intrigued by it because, and this is getting to your point earlier, Heather, and totally cut this because I might be going off topic, but you were alluding to putting stem cells into different spaces. And whilst I agree that it has a reduced capacity to probably do something if you put it into the wrong place, I also think we don't fully understand the ways in which stem cells contribute beyond their niche as well. So it was always considered that the stem cells within fat. So when I talked about how fat cells, are made from a progenitor, made from a stem cell, they're largely considered to exist within the fat tissue, lining the walls of the blood vessels and giving rise to their progenitors that sit in the adipose tissue. But a study that I was involved in using patients who had actually been having total body irradiation for their blood cancer, and then years later, these patients came back and they were surviving from 3 years to 17 years beyond their cancer. And then they came back and gave adipose samples. And we isolated fat cells and took the DNA out of individual fat cells. And you could match the DNA to whether it was from the patient or from the donor, because these patients that received the total body irradiation had bone marrow transplants or peripheral blood mobilized. Yeah, anyway, they had basically bone marrow transplants. And so you could actually see that there were fat cells that had the DNA that was from the donor, from the bone marrow 15 years earlier. So these bone marrow stem cells were able to go into circulation, and even more interestingly, in the individuals with obesity, they had more of these. So it seemed like mobilizing these stem cells to the adipose tissue was actually able to contribute to the growth of the adipose tissue in obesity. So I also think there's a lot there that we don't understand in how the niches talk to each other, the plasticity within the niches, where to put them, when to put them, etc.
Yeah,
I mean, that
and I
sounds
think
like
that's
some
this is
sort
one of the things
science
with
fiction.
but with mesenchymal stem cells, I think the two areas that it is well defined that they're able to differentiate, self-renew and differentiate, is adipose and chondrocytes, like cartilage. So there's definitely sort of opportunities there. I don't mean to poo-poo mesenchymal stem cells, the poor buggers.
And when we think about chondrocytes, right, those are the cells that secrete that help your cartilage, right? So when we think about knee and shoulder and these joint issues, I mean, that's actually really exciting for people.
They've been carbon dated as well, and they don't really turn over.
Bummer. I'm just thinking of like my old knees as I'm starting to get the aches and pains of age. That wow, that would be awesome if I could just get a stem cell shot.
They don't endogenously turn over, but maybe Heather one day will solve that and be able to transplant them in there for you.
Or
someone will work out how to make them endogenously turn over.
So I guess since I have the two of you on, what can we expect out of stem cell therapies? I know it's kind of a crazy question, but what's promise? What's hype? What, as we particularly talk about health span, right? And the things that kind of affect us with age. So brain function, musculoskeletal strength, right? Not dying of cancer, not dying of cardiovascular disease. What are some of the things that stem cells show promise for, like real scientific promise, and what are areas that just seem a bridge too far that we may not be able to reach kind of within the next 20 or 30 years based on our understanding today?
I'm going to take first one, Heather, because it's an easy one.
There you go for it.
That's not an intervention that everyone needs to wait for or be rich for. Exercise. That is one thing that has been shown to promote neurogenesis, say, for example, in the brain. So it clearly does something. It engages with stem cells. It increases activity. So there are things that and I'm sure there's more things, and maybe, diet for sure, increased with obesity and lipid can inhibit stem cell activity. So certainly taking care of yourself in terms of diet and exercise can keep your stem cells healthier.
Yeah, and that also then kind of feeds into the concept. If things like diet and exercise can keep the cells healthier and younger, then certainly there's going to be interventions that could do something similar as well. I know we're all lazy. We all want, I'm not going to say the names of certain medicines that we would prefer to have than do exercise. No, so I think it defines it quite well in terms of the potential lifestyle changes, the mechanisms that are going to try to play with that endogenous ability to renew and regenerate, and then the mechanisms that are going to supplement where that endogenous ability is lost.
No, I think that's awesome because I focus a lot in my clinical practice on diet and lifestyle interventions. And I always say the most powerful pharmacy we have exists inside of our bodies and our cells already. So exercise is one. What are some of the other dietary or lifestyle things that may be able to help our stem cell population, take care of these precious cells? Because I think in the integrative functional universe, there are a lot of plant-based supplements that they say boost your stem cells, and I don't know what's hype and what's not, but I certainly think that exercise I love. Calorie restriction, I think, has been something that they talk about as well. So like intermittent fasting, as being able to help with autophagy and boost somewhat stem cells. So are there other things that you guys can point to that may help our nascent, precious little stem cell population in our bodies that we can do on a daily basis?
I got nothing.
Nothing.
Well, senescence is a term where cells that are normally cycling kind of exit cell cycle and they stay stalled, so they don't die. They become very metabolically active. They secrete a lot of things that
They're called zombie cells and SASPs, right? The
yeah, they have the
secretory associated
senescence associated secretory phenotype.
something like yeah.
And there's lots of promise in the terms of like senolytics and xenomorphics, they're called, that clear these zombie cells, if you will, these senescent cells, and dasatinib and quercetin, the resveratrol. So there certainly are, probably certain foods that are higher in these components. I mean, I always grew up on an apple a day keeps the doctor away, and I think that's high.
It's true.
In question. So, I think that there is a lot of really cool, fun science going on where people are really looking at the pharmaceutical actions of many of the different fruits and vegetables that we should be eating, and particularly for this. And so I think, that in terms of, there was shown that senescence in the progenitor cells in the adipose tissue, so sort of these zombie cells in fat, they kind of stop the ability to make new fat cells, which means the lipid gets put into the existing cells. And half of the health consequences of obesity is not how much fat you have, it's where is it and how big are the fat cells. So if you have premature aging, this senescence and this zombie-like state, if you will, in the progenitor cells, the stem cell derived sort of progenitor cells, they can't do what they need to do. They can't make more fat cells. And ironically, you'd think that's a good thing, but it's not, because the lipid will then go in to the other cells, and either the existing fat cells will get really big, which means they stop functioning well, or the fat cell will not take the lipid, and then it will go to what we call ectopic sites. It'll go to the muscle or the liver or the pancreas, and it will definitely impact on their function. So you really want operational fat. You want the ability to make new fat cells because it saves your body from lipotoxicity and everything else.
Wow.
Having senescence in the progenitor cells is a really bad thing.
That's a mic drop moment.
Yeah, I was going to say, I think our next episode might be senescence, Sanjay.
No, I think that's great. And is there a difference in these adipose-derived cells from visceral fat versus subcutaneous fat, right? We think of the pinchable muffin top type stuff, right? And I can speak from my clinical experience where I think visceral fat is much more metabolically active. There's a whole new science of adipokines and disrupting gut to brain signaling. And I tell people, lovingly that the visceral fat is the fat that suffocates our organs. But I think there's a whole new kind of realm of study of this. So as we look at these adipocyte-derived cells that we need more of, I guess, to get to, so that we're not limiting our cell, our progenitor cells and adipocytes. Is there a difference one versus the other, or is all fat created equal? Or do we know?
No, definitely not all fat is created equal. And certainly when it comes to clinical correlations with adiposity, with fat, having it on the thigh, the superficial fat that's on the thigh and the buttocks and the belly is all good, basically. Having it around the organs is pretty much bad. And that's where all the correlations come with, cardiovascular, type 2 diabetes, etc.
Right.
So that holds. But senescence, when we look at the mature fat cells, we don't see it in the visceral fat. We see it in the subcutaneous fat. But when you look at the stromovascular, so where the progenitor cells, and back to the sort of the stem cell discussion, then it's been shown in the visceral fat. So you then maybe get more expanded out white fat cells there, and you get more fibrosis. And this is a very negative cascade of chronic inflammation, which is very bad systemically. But traditionally, yeah, the omental, the visceral fat that sort of feeds, via the hepatic portal vein more directly into the liver. So it dumps more things into the liver, more lipid into the liver. And we know that fat in the liver is the beginning of all kinds of
Nonsense.
unbalance. And yeah, so it's really,
Yeah.
you want to protect your liver from this. So a mental fat and the visceral fat, like you talked about, it definitely is very different from the other fat depots. We don't fully understand exactly why. There's lots of theories, and we know that they behave very differently. In my experiments, we definitely see senescence is different, but I don't think we're not completely there yet in understanding why. And I don't think that subcutaneous fat is great. I think we also don't appreciate the pathology that goes on there. We generally just see once you get your subcutaneous fat so full and big, it then moves over to the visceral fat. So when you start to see correlations, yes, it correlates with visceral fat, but the subcutaneous fat has sort of stopped working in order to get the fat into the visceral fat.
So
Okay.
there's an adipose dysfunction. There's a fat dysfunction.
There is. You can give
Wow.
drugs. Ironically, one of the type 2 diabetes drugs, the TZDs, thiodiazine.
Thiazolidinones. Yeah.
Thank you. I'm glad we got the MD in the audience. So they,
Exactly.
ironically, they made the patient healthier. They used to be trialed for type 2 diabetes. They would make the
Yeah.
person healthier, much better, like blood lipid and glucose profile, etc. But it made them fatter because what this did was push the progenitors to make more fat cells. So their people ended up expanding out their fat, which was healthy because they now have more small fat cells.
Right.
So all the lipids going into the right place because fat cells are designed to store lipid. Your heart, your pancreas, your liver, your muscle, that's not designed to function with lipid all around it. So the
Yeah.
moment you start absorbing all the fat into the good cells, it's actually healthier,
Huh. Boy,
which
I
is,
have like so many questions. But I think we'll have to reserve them for our next episode. I mean, this is fascinating. Boy, we started at nuclear testing to brains to hearts to now fat cells, and there's different types, and we want more fat cells, not less. I mean, it makes me wonder what happens with liposuction, and boy, all these clinical questions I have, right? Like, so the point is, though, I guess apple a day, sleep as much as you need to, exercise by lifting heavy things and walking for longer than you think you should, and those things kind of help our bodies.
yep. I mean, it's all
Uh,
common sense things, but we're putting some science to it. It's,
yeah, I call it grandma medicine. Do all the things your grandma told you, right? Like go outside, play until it's dark, eat your vegetables, don't be mean, all the things, right? Like have a lot of friends. I think that's great. Well, boy, Kirsty, so much learning. I would love to have you back and kind of dive into kind of some of your more specific research, your zone of genius things, because I think the adipocytes, there's so much we know and so much we don't know. And I always say the farther you swim from shore, the deeper the water gets. So the more we think we know, the less we actually know. So I think it's just fascinating. Heather, why don't you take us from here?
I think that's a great way to say that's why we're here, so we can know less, right? We can know more and realize that we know less. I think this was really fun, and it was so great to have you, Kirsty. Thank you for joining.
you're welcome.
And yeah, I think we will try to bring up some senescence in the coming episode, and we'll try and get Kirsty back another day to talk about the adipose research. I think it's been fantastic.
Yeah. So thank you guys for listening to this episode of the Healthspan Action Coalition podcast. If you like what you heard, please subscribe. We love getting more people that love us, right? That's so we love to grow. And if you have questions, please put them in the comments. And if you have someone that you think that we should interview or chat with, let us know. We'd love to expand our network and bring like-minded scientists, clinicians, people from industry who are trying to advance the science of health span in their own way. So leave us a comment, and we'll catch you guys next time on the Healthspan Action Coalition podcast. Bye, everybody.
Bye.
Bye.